Hi everyone! Today we're going to be talking about T-cell activation and effector function. Before we begin, I just want to remind you that all of the figures from this presentation are coming straight from your Janeway Immunology book, so you'll have the opportunity to follow along. However, this lecture, unlike some of the other lectures, will jump a little bit between chapters, so make sure you pay attention to the bottom left of your screen. There you'll see little numbers that come up that will indicate the chapter from which the figure is appearing, and also the number of figure that it is within that chapter. So to start today, I'm going to first outline our 4 main objectives. This will give you a sense of what to expect moving forward throughout the lecture, but also a sense of what you should take away from the lecture after we've finished. First, we're going to introduce secondary lymphoid organs and sites of lymphoid activation. Then, we will learn about lymphocyte homing and migration through secondary lymphoid organs and important chemokines and surface molecules. Third, we will learn about the 3 necessary signals needed to activate a T cell. And finally, we will discuss different co-stimulatory molecules. So, to begin, we will first talk about the secondary lymphoid organs in our bodies. You've probably already learned about the primary lymphoid organs, the bone marrow and the thymus, where lymphocytes develop. However, once the lymphocytes are mature, this is not actually the place where they become activated. Instead, they become activated in a set of secondary lymphoid organs. These include the lymph node, the spleen, and the Peyer's patches. Lymph nodes are located all over the body, whereas there's only one spleen in our body. Peyer's patches are located in the gut. I'm not going to speak too much about Peyer's patches today, in part because they mimic the structure of the lymph node, but also because the biology of Peyer's patches is much easier to understand in the context of gut immunology and the microbiome, which you'll hear about in later lectures. So let's start with the spleen. The spleen is located right along the route of circulation as the blood circulates from the heart around the body. Because of this, it traffics both red blood cells and lymphocytes. The spleen is split up into 2 sections, the red pulp and the white pulp. The red pulp is where red blood cells will travel through and go to die when they are getting old or are damaged. The white pulp is where lymphocytes go. Lymphocytes will enter through the central arteriole and then separate into either the B cell zone or the T cell zones. There, they will encounter specialized dendritic cells that can show them antigen and potentially activate them for an immune response. The nice thing about the spleen is because it is so centrally located, it is the perfect spot for the adaptive immune system to become activated in response to blood-borne pathogens. In contrast, there are several lymph nodes around our body, and these are all associated with peripheral tissues. This means that if a tissue like the liver, the lung, or the kidney becomes infected, dendritic cells in that tissue can carry the pathogen or parts of the pathogen, antigens, to the lymph node where they can activate an adaptive immune response. Here, similarly to the spleen, the T cells will enter through a vessel called the high endothelial venule, where they will then separate into their respective B cell follicles and T cell zones, where they will encounter dendritic cells that can then tell them whether they should activate an adaptive response or not. Then they will leave through the medulla. So how does a lymphocyte even enter a secondary lymphoid organ once an infection is found in the body? This brings us to our second objective for today, which is to learn about lymphocyte homing and migration through SLOs, as well as the important chemokines and surface molecules involved in this process. So leukocyte homing, or lymphocyte migration to the infected tissue, occurs in a process called leukocyte homing that can be split into 4 separate stages. These stages are rolling, in which the lymphocyte will stop flowing freely through the blood vessel and will instead begin to roll along the endothelial layer that lines the blood vessel. The second stage is activation. Activation occurs using a specialized type of cytokine called a chemokine. The third step is adhesion. adhesion. At this point, the cell will stop rolling along the endothelial layer and instead will halt. This stopping of the cell will allow it to migrate through the endothelial layer in a process called diapedesis. So first, let's start with the rolling. Rolling is mediated by molecules on the T cell surface called L-selectins. L-selectins bind to molecules on the endothelial surface like CD34 and glycam-1. This interaction stops the lymphocyte from flowing freely through the blood vessel and allows it to start rolling against the endothelial layer. This first step is important because it slows the lymphocyte down enough that it can start receiving the activating signals that we see in our second step here. So during this step here, activation, the lymphocytes rely heavily on signaling through by a chemokine called CCL21. We'll talk a little bit about chemokines in later lectures. For now, what you need to know is a chemokine is like a traffic signal. It tells the lymphocyte it needs to get ready to get off the highway and where to go when it does. CCL21 does many important things in this scenario. First, it's an important signal for T cells, guiding them to the lymph node and eventually to the T cell zone within the lymph node. It's produced by high endothelial venule cells, lymph node stromal cells, and dendritic cells in the lymph node. All of these different cells producing the chemokine allow it to form a gradient. This means that CCL21 will become more and more concentrated the closer the T cell gets to the lymph node. This way, the lymph node knows where it should go when it leaves the blood vessel. CCL21 signals to T cells via the receptor CCR7. This first interaction between CCL21 and CCR7 on the T cell is what activates the T cell for our third phase of leukocyte homing. This brings us to adhesion. Signaling of CCL21 through CCR7 tells the T cell that it needs to start upregulating a molecule called an integrin, like LFA1. LFA1, expressed on the surface of the T cell, is able to interact with adhesion molecules on the endothelial cells. One of these adhesion molecules is ICAM1. These adhesion molecules have different names based on where they're expressed, but for now, let's just focus on the LFA1-ICAM1 interaction. The interaction between LFA1 and ICAM1 actually stops the lymphocyte from rolling and instead halts it on the endothelial surface in the blood vessel. Because the T cell has now stopped rolling and instead is sitting still and stationary on the endothelial layer, it is able to migrate through in the process we call diapedesis. After diapedesis occurs, the T cell can continue following the cytokine CCL21, as well as the cytokine CXCL12, which will guide it to the lymph node that's associated with the tissue that's been infected. So let's go over this together one more time. First, a circulating lymphocyte will enter the high endothelial venule, or the highway that will eventually get it to the lymph node. Binding of L-selectin on the surface of the T cell to glycam-1 and CD34 on the endothelial surface will stop the lymphocyte from flowing freely through the bloodstream and instead cause it to roll across the endothelial surface. This rolling allows CCL21 to signal through the receptor CCR7 on T cells. This signaling will cause the T cell to upregulate the expression of the integrin LFA1. LFA1 will then interact with adhesion molecules on the endothelial surface, like ICAM1, that will then stop the rolling motion and cause it to halt completely on the endothelial layer. Once the T cell has stopped, it is able to migrate through the endothelial layer in that process called diapedesis. Then it will traffic on to the lymph node. Once the T cell gets to the lymph node, it will see a dendritic cell. The dendritic cell will eventually present antigen to the T cell, but in order for that to happen, the T cell and the dendritic cell must first establish an interaction. This interaction is established using some of the same molecules we just talked about that the T cell uses to get into the lymph node. One of them is LFA1, which you'll remember is an integrin that interacts with ICAM1 on the surface of the dendritic cell. This first initial interaction allows for a low affinity relationship to form between the dendritic cell and the T cell. Because of this, the T cell is now able to have its TCR meet with the MHC presenting peptide on the antigen presenting cell or the dendritic cell. This secondary interaction will tighten up the first interaction between LFA1 and ICAM1. This together will create a prolonged interaction between the T cell and the dendritic cell. This will give the T cell time to decide whether it recognizes the antigen that the dendritic cell is presenting or not. This brings us to the decision that the T cell must make upon interaction with the dendritic cell. It will do one of two things. Either the T cell will not recognize the antigen being presented by the dendritic cell and will leave the lymph node via the cortical sinus. Alternatively, the T cell may recognize the antigen that the dendritic cell is presenting and lose its ability to leave the lymph node and instead start proliferating. You'll remember from previous lectures that there is a large amount of diversity in TCRs and in our T cell repertoire. Because of that, there's actually a very low number of T cells that will recognize any given antigen presented by the dendritic cell. So this means that 9,999 cells out of 10,000 will not recognize the antigen presented by the dendritic cell and will instead leave the lymph node and go on their merry way. 1 out of those 10,000 cells, however, will recognize the antigen presented by the dendritic cell and will stay mounting the adaptive immune response that will protect you from infection. So how does the T cell even become activated? Well, that brings us to our third aim for today, or our third objective, which is to learn about the 3 signals necessary to activate a T cell. However, before we talk about these 3 signals, we first need to understand how the dendritic cell, the cell that actually presents the antigen to the T cell, gets to the lymph node with the pathogen. Dendritic cells are located in the peripheral tissues where they can see the pathogen that could infect the tissue. Dendritic cells can be activated by innate immune signals that you would have heard about earlier. This includes signaling through TLRs or other pattern recognition receptors. An example would be LPS, which is a component of gram-negative bacteria that would signal through TLR4 on the dendritic cell and activate it for immune response. These innate immune signaling responses are necessary to activate the dendritic cell such that it knows it is ready to take up pathogen and migrate to the lymph node and activate T cells. Once the dendritic cell has received its innate immune signaling, it will begin to migrate towards the lymph node. This occurs by upregulation of the cytokine or the chemokine receptor CCR7. You'll remember CCR7 from our talks about lymphocyte homing. CCR7 recognizes CCL21, which is being produced by the lymph node. CCR7 on the dendritic cell will allow the dendritic cell to follow the same gradient that T cells are following from the blood vessels to get to the lymph node. During this migration, other things are also happening. The dendritic cell is upregulating the expression of co-stimulatory molecules called B7 and the MHC molecules that will present the antigen. Both of these things become essential for delivering the 3 signals that T cells need to become activated, so keep them in mind as we move forward. Once the dendritic cell gets into the lymph node, it can start presenting the antigen to T cells. It may also hand off its antigen to other dendritic cells that are already located in the lymph node. These dendritic cells will help share the load for presenting antigen to T cells.